Welcome back to the study of enzymes. Now we are going to see the structural details and organizational details of enzymes. Basically, enzymes may be of two types as far as their organization is concerned. Some of them are very simple enzymes because they are consisting of only protein molecule, while others are complex enzymes or holoenzymes because they are associated with a non-protein part. And that non-protein part is called a cofactor, while the protein part is called an apoenzyme. Remember, in this topic, we are considering only the proteinaceous enzymes. But remember, all enzymes are not proteins. Never believe that all enzymes are proteinaceous in nature. When we study the proteinaceous enzymes, these factors are applicable. So, cofactors are the non-proteinaceous part of the enzyme. This cofactor may be either a prosthetic group or it may be a coenzyme. When is it a coenzyme? When it is an organic molecule and when it is loosely bound to the apoenzyme, when it is transiently bound to the apoenzyme. And then what's a prosthetic group? A prosthetic group is often a small inorganic molecule. It may be an atom also, and it is, look here, it is tightly bound to the apoenzyme, permanently bound to the apoenzyme. And remember, there are some mineral ions which are cofactors but which are not coenzymes. We will see them in the following slides. Look at the diagram here. Apoenzyme is the proteinaceous portion of the enzyme, of that complex enzyme, but it by itself is inactive in nature and it is associated with a coenzyme. What's a coenzyme? Coenzyme is an organic molecule, but it is not proteinaceous in nature and it is an activator. Now you see, this is the apoenzyme, this is the coenzyme. Together they make up the holoenzyme and the holoenzyme is active. That holoenzyme is going to interact with the substrate. It is going to act upon the substrate to convert it into products. This is a paragraph taken from a textbook. Whatever I told you right now is given here. Can you just find out the name of the book and the page number? It's actually famous. I have given you this book. Whatever I told you already is given here. You see, some enzymes require additional chemical compound called cofactor. Sometimes they are inorganic ions. For example, ferrous ion, manganese ion, magnesium ion, manganese ion, zinc ion, etc. are there. Sometimes they are organic in nature. We call them coenzyme. Okay. So all those things are given here. This is a diagrammatic representation of whatever I told you right now. Simple enzyme, conjugate enzyme, apoenzyme, cofactor, etc. are there. Now, remember, all enzymes are not proteins. For example, ribozymes. Ribozymes are RNA molecules that catalyze reactions on other RNA. You might have studied all of those things in your previous classes. Again, we come to cofactors and coenzymes. Cofactors are required for the activation of some enzymes. They may be ions or coenzymes. While coenzymes, they are complex organic molecules, and so the coenzymes are also cofactors in nature. They may be vitamins, they may be derivatives of vitamins or as molecules. NAD is a good example. You might have heard of B complex vitamins. When there is a deficiency of the B complex vitamins, we may get some deficiency diseases in our body. Most of these B complex vitamins are actually coenzymes for some enzymes. So their function is to act as coenzymes. Therefore, We should get these coenzymes in our diet because these coenzymes are obtained from vitamins and they are essential organic substances, and we are not able to synthesize these vitamins in our body, so we should get them through our diet. Examples, thiamine, niacin, etc. They are functioning as coenzymes. Here is a list of some mineral ions required for the functioning of the given enzymes. Very much important. Reference is given here, Leninger Principles of Biochemistry. I told you some mineral ions are cofactors. You see this enolase is the enzyme. This is the whole enolase molecule, enzyme molecule. It has two magnesium 2 plus ions as cofactors. So this is the enzyme molecule and these are the cofactor molecules. Here some coenzymes. Coenzyme A. Coenzyme A is actually a vitamin called pantothenic acid, derived from pantothenic acid, and it's a carrier of acyl groups during chemical reactions for the acyl transferase enzymes. Biocytin is a coenzyme obtained from the vitamin biotin. It's a B complex vitamin and it is a carrier of carbon dioxide. Like that. In this column, you have the different vitamins. You can see vitamin B1, B2, B6, etc., etc. B12 is cyanocobalamin, vitamin B12. And these are the coenzyme forms of the corresponding vitamins. And when they are acting as coenzymes for enzymes, what are the functions of this coenzyme? They are transferers, they are group transferring molecules, these coenzymes. It's a very important section. Reference is given here. And again, some vitamins, some of them are shown in the previous slides also. They are coenzyme form, the reactions they catalyze. And here, the important fact is that example of reaction is shown here. You keep a note of it. It may be asked for entrance examinations, etc. Examples of the reactions being catalyzed by the enzymes where nicotinic acid or vitamin B12 or vitamin B1 or pantothenic acid, etc. are coenzymes. This is another one. You see, nicotinamide adenine dinucleotide is NAD. NAD is a very important coenzyme and it is acting as a hydrogen donor or recipient in dehydrogenation reactions. You might have heard of the class of enzymes called dehydrogenases. It's given here also. And NADP. NADP is meant for specific dehydrogenases because that's also carrying hydrogens or protons. Better we say hydrogens. FMN, FAD, etc. are being acting as coenzymes of different enzymes. You might have heard of succinate dehydrogenase in Krebs cycle. You know that FAD is the coenzyme for that enzyme. So make a note of this slide as well. Now coming to prosthetic group, I told you earlier that prosthetic groups are tightly bound molecules. They are organic molecules. The prosthetic groups are tightly bound to cofactors. They are organic molecules. Tightly bound with what? Tightly bound with the proteinaceous part of the enzyme. So this is the apoenzyme without the cofactor. Now the cofactor is going to bind with it in a tight manner. When it is binding tightly, we call it a prosthetic group. Binding tightly means what? Using covalent bonding. Now this assembly is actually the active holoenzyme. This is the active holoenzyme. Now the enzyme is activated. It is ready to act upon the substrates. Okay. In peroxidase and catalase, heme is the prosthetic group. Heme is the prosthetic group. Sometimes you might have studied HAEME as the spelling. Don't worry. Difference between American English and British English. Okay, no problem, don't worry about it. This is the molecule. This molecule is actually hemoglobin. It is there in your blood. This hemoglobin is having heme as the prosthetic group. But we are studying about enzymes. hemoglobin is not an enzyme, then is the term prosthetic group applied to non-enzyme molecules also? Yes, the term is applicable to all proteins. And of course, many of the enzymes we study are proteins. And the protein prosthetic groups, non-protein components, for example, metal ions and iron, magnesium, etc. All those things I told you earlier. Heme is there. This is the structure of the heme molecule. Here iron is there. Fe2 plus ion is there. And again, another side of hemoglobin. Each subunit of hemoglobin consists of a globin, a polypeptide chain, and a heme. Of course, these things are already given to you. Hemin, you might not have heard about this. Hemin, beta hemin. Peroxidases from food of plant origin and several catalases contain ferriprotoporphyrin. This ferriprotoporphyrin is called hemin, and it is the prosthetic group of those enzymes. They are the chromophores. Chromophores means what? A part of a molecule which is providing it with a certain color, because it is absorbing light of a particular wavelength in the visible light region. That's why it's having color. And again, prosthetic group of some other enzyme. You can see here carbonic anhydrase. What is carbonic anhydrase? You know that carbonic anhydrase is responsible for conversion of carbon dioxide and water into carbonic acid, and it will help transport of carbon dioxide from the tissues towards our lungs much easier. And carbonic anhydrase is one of the fastest enzymes known to us. It is a zinc enzyme. Carbonic anhydrase is a zinc enzyme because it contains firmly bound metal ion. Which ion? Zinc. In this case, it is zinc ion as the prosthetic group. Okay, only this much.